Good morning, everyone, and welcome to the Elixir Friday Coffee, where we're presenting tools and services which are provided by the infrastructure ELIXIR-CZ. So today is a time to show the very successful implementation of some software developed years ago. And Karel Berka will be talking about Mole Online, which is a great tool for, I suppose, still searching for the cavities in the proteins. So Karel, the floor is yours.
Good morning, ladies and gentlemen. My name is Karel Berka, and I will be speaking about the Mole Online, which is our tool for the analysis and calculation of channels, tunnels, and pores in biomicromolecular structures and storing them in channels DB. And you might ask, why do we study channels, and why would anyone study channels? And the major reason is that almost 2/3 of enzymes actually contain a channel leading to the catalytic side. And as you can see from our picture from our paper of anatomy of those enzymes and the tunnels, meaning that the composition of the channels is quite different and full of aromatic amino acids in comparison to what the catalytic sites look like and what the interior of the protein or the outerior of the protein looks like. So it's a distinct feature which allows the enzymes, as Jiří Damborský is saying, to add a key lock into a key and lock model for the enzyme function. So it probably allows for the substrate recognition as well. And the second reason why it's important is that the transporters and channels and pores are quite important for membrane transport on drug actions. On the right side, we have a selection of what type of the drug targets are in Campbell. And as you can see, there is quite a lot of those stuff are already important for the drug designing. So we've developed years ago a Mole Online, which is available at mole.upol.cz. And this is our tool for the detection and analysis of channels and tunnels. So upon the uploading the structure, either from the PDB or from your own structures, as we will hopefully see at the end, you will see the structure, and then you can calculate either pore or channel. And the pore, I mean transmembrane pore, because we calculate also the position of how the protein is positioned in the membrane, and therefore it allows to guide the analysis of the transmembrane pores. Upon the pore is calculated or channel, you can select it, and from the selection you will see the profile of the channel and its properties, like different charges, hydrophobicity, polarity, mutability, plenty of other physical chemical properties. And in the latest version, our tool is able to read not only PDB files, but also MZIFs. And it can calculate multiple properties which are quite common for small molecules like log P, log S, so solubility and partitioning to the membranes. The tool is also combined with the channels DB that is already added into the PDB-E knowledge base as well. So any calculated channel that is annotated by this tool is then sent to our team, which verifies it and put it into the database that is afterwards mirrored to the channels to the PDB-E knowledge base and available to the general public. And here on the right side, you can see the differences between the closed and open channels that quite nicely shows where is the charge selectivity filter in this triple ML porine structure. The channel annotations are taken from the literature, or we are automatically calculating them from catalytic sites and enzyme cofactors. And these are usually calculated not only with the online version, but we are using our command line app that can be quite useful for the large-scale calculations, because our tool is the quickest one on the market to calculate the tunnels. So now I will show you a short showcase about how we calculate these things. So here I've uploaded the one-take QNs, which is cytochrome P453A4 protein, and I will select by some kind of origin from where I want the channels to be calculated. I've selected this path, add it as a starting point, and submit. And now we are calculating online, and you can see that we have 3 different channels already. Those properties are shown here, and you can, based on those properties, we can basically try to understand how these things are working. And you can see that the first one is the smallest, and it's closed tunnel in that way. And there are 2 tunnels which are quite similar in their properties and their openness. And since we now have structures in for basically any protein from human in AlphaFold database, then we can look into how this protein look like in AlphaFold database, and we can see that it's quite similar. And since I've downloaded it and uploaded it to the mole online, here is the structure, and we see that basically what we calculated is similar set of the channels. So this is a word of warning that the channels are opening and closing based on how the proteins breathe. So that's quite important when you are analyzing molecular dynamics trajectory, that the channels can be open and closing during the time, and that's the reason why we developed the command line application as well, to help you with the analysis of this. So here the channels are, the open channels is here the same, and the solvent channel is here analyzed. And since the transmembrane alpha helix is shown in the AlphaFold database like this, and it's quite similar to what our simulations were showing us all around the time, we can basically say that these tunnels are leading towards the membrane, and they are sucking the substrates, and this channel, which is closed in apostructure or more closed, is apocluster. Therefore, the solvent channel and the channel for the product release out from the membrane is actually closed in this structure. Similarly, we can analyze the pores, and for the pores, I have another case, and that's the ATP binding cassette transporter ABC1. And this transporter is responsible for the drug transport from the cell outwards. And there is no crystal structure for it, so I've taken the AlphaFold. And for this structure, I've calculated the pore. So here I've switched towards the pore model. And you can see that the structure is positioned in the membrane by uses of membed prediction algorithm. And we can see that the road through the protein is basically leading to this cavity in the center, and then the rest of the channel or pore is basically quite small and closed, and that's actually so small that the drug would not cross. But upon the transport cycle, when the ATP binds into these so-called NBD domains, those NBD domains will close each other, and the structure will open towards the extracellular domain. So, but from what we see here is the way how the molecule and where the molecule would reside in the pre-transport cycle. So there were the two examples, and I would summarize that the mole online is quickly calculating channels and transmembrane pores online. It can provide the analysis on the calculated channels in the means of their properties. It's connected with the channels DB, so it gathers the channels from mole online. And in mole online, and that's the thing which I forgot, is that here I can select also what channels are calculated within the channels DB. And for those, we have the annotations automatically downloaded from the channels DB. So we can compare quite easily how these channels are similar or not similar to what we have from the previous calculation, from previous analysis, which are stored in the database. And you can also use a command line to run the thing. With that, I need to acknowledge my collaborators and especially the late Professor Jaroslav Koča, who unfortunately passed this summer. And with that, I would like to thank you for your attention, and I will answer any questions.
Thank you very much for very interesting intro to the mole online. There are a few questions now. Some of them actually came from me. So I'll start it. The ask. I suppose that most of the structures you are analyzing come from the X-ray. So how much the identified cavities could be influenced by crystal packing forces?
Well, it certainly depends on where the crystal packing is in comparison to the catalytic site, for instance. I know the structure of the cytochrome P450 2D6, which is the PDB code is 1P05, and there the crystal packing is actually locking inside the catalytic site because it basically the structure is showing two structures very tightly bind into each other at catalytic site. And in that case, it definitely changes the crystal packing changes how the channels will look like at the end. But that's a quite strong artifact. Normally, if you have, I would say, typical level of the crystal contacts, those are on the sides and usually not in the spots where the channels or the catalytic site is.
Yeah, Marian is actually following my question. So he asked if you look at the similarity of results for different structure of the same proteins.
Yes, we did, and we will probably rerun this task again because there is quite a lot of new structures, and not only in the AlphaFold but also in the experiment since we've done this analysis. And what we found is that the variation is quite depending on the ligand which is in the structure. Because if the ligand is large, then it inflates also the channels leading to the active site. So the apo structure usually has quite small channels, and those channels can be opened by the ligand which is passing. So the variation can be quite big, and it's important therefore to look for the channels that are staying in the structures or that are opening by the movement of the ligand.
But in principle, it means that even the channels or tunnels are quite flexible upon the ligand or probably external forces, right?
Yeah, sure.
Vojta is asking if you because you presented running mole on AlphaFold results, and the question is if you plan to do that systematically on all AlphaFold model proteins, how they are accessible now from the EBI.
Yes, that's our plan.
Okay, so I suppose that Vojta is satisfied. One more question. So you said that most of the tunnels are located in enzymes, or 65% of them. So how you interpret the tunnels in non-enzymatic proteins? What are they for?
They usually, in that way, they usually are for the movement of the ligands inside the structure. So, for instance, here in this KDP Fab-C complex spore, there are actually two tunnels, where one tunnel is for the interactions from the outside, and then there is a specific tunnel in between those two chains, where the molecule of water is passing from one side to another to change the conformation on the other side by just this movement. So it can have this type of the activities at the end.
So we can look on the tunnels as the tunnels for water or for solvent?
Yeah, sure. That's actually quite typical. And here, ribosome is not enzyme as well, and we can analyze the ribosomal exit tunnel as well. So
regarding the ribosome, so this is quite flexible, right? And it actually depends on some cofactors which are bound to the ribosome. So how much, for example, is changed the tunnel in the ribosome?
Well, in case of ribosome, you need to change quite a lot the entering parameters, because normally the protein which is generated by the ribosome is quite large in comparison to small molecules which we are analyzing, for instance, in the case of the enzymes. But by tuning the parameters, it's possible to look into the to analyze the ribosome and then to compare it in between the individual structures. But since we are not really interested in ribosomes, we did not done this analysis further, only by just looking that we are able to do it.
Okay, and probably the last technical question: can we implement the mole online locally, or is available the code? You can actually run it locally?
Well, you can definitely run the command line application.
Okay. So I don't see any other question. So Karel, thank you very much again for the very, very nice talk and the introduction of a very, very useful tool. So thanks to all who contributed to the discussion, and see you on next Friday at the Elixir Friday Coffee. So have a good day and bye.
Yeah, bye.